Hello and welcome to this video on a means of producing artificial blood. One donor can give 600 milliliters, or 1/3 of a unit of blood. This can produce up to 27 products or be transfused into a single patient. Considering that one car accident needs an average of 100 units of blood, this is a relatively small number per an individual donation. It is also an acute need, and when compared to that of surgical patients, cancer patients, and those with chronic diseases, it is an insufficient amount. In the UK, 1.5 million units of blood need to be collected annually to meet the needs of patients. This is a largely inefficient process, and donors must be matched to recipients, and even then, further refinements are necessary to accommodate individual variances in blood type and immunological responses. The most common type of these is type O, which is also the rarest. Between 3 and 7% of most populations donate blood, and considering type O can make up a very small proportion of this is a limited pool that is subject to seasonal and event effects such as influenza and public holidays. Being able to both supplement, if not replace donation via a synthetic process is something of a holy grail. It would be used to smooth out the demand and supply issues of blood donations. An example of the need for this is the events in Florida, United States of America, in which after a mass shooting there was a depletion of blood supplies across the state, so much so that there was not enough blood available for treatment of those who were injured. Although eventually solved, this combined with seasonal effects leads to a degree of instability in the supply chain. Through years of research and development, this is now on the horizon, although not yet at the point of ultraviolet. Other than a high demand for blood products, synthetic blood has a significant number of advantages over general blood products. This includes a reduced risk of disease transmission and host rejection. The cause for this achievement is based in two fields. The first is the ability to use and understand how immortal cells work and where they came from. The second is the ability to differentiate stem cells into a known product, this being the overlapping of molecular cell biology and oncology. First things are to explain what immortal stem cells are. Stem cells are cells which can change into another cell when they undergo mitosis. This is seen in the replacement of red blood cells, all of them, red cells, immunological cells, and platelets. This is through a cycle of creating one new cell that goes into differentiation, and another which remains and undergoes the process repeatedly. At least that is how it works in humans. The how and why of this is complicated, but in simple terms, a series of timed signals interact with surface markers to activate key gene pathways. This causes the cell to build and to modify the cell structure, and consequently function. This is done in the bone marrow under controlled environmental conditions. Immortal stem cells are a hybridized stem cell that is being combined with a cancer cell line. The reason for a cancer cell line is the ability to undergo meiosis and mitosis indefinitely. This creates a cell line that will not die due to the number of times it divides, unlike regular cells, which have a finite number of replication cycles. In the case of blood cells, this means it can continuously produce cells, which would be a problem in the case of cancer like leukemia. This is where the new development comes in and why it is such a significant breakthrough. Past research has attempted to turn donated stem cells into mature red blood cells. This was highly experimental and incredibly inefficient. It was therefore unsuited to mass production of blood. Each stem cell could only make 50,000 red blood cells before it died. This is the point at which the researchers needed a new blood donation with new stem cells. Meanwhile, 50,000 cells might sound like a lot, but putting that into perspective of a typical bag or unit of blood used in hospitals, there are 1 trillion red blood cells in there. This is less than 1/100,000th of a bag of blood, or less than 1,000th of a blood donation, nowhere near sufficient and nowhere capable of supplying the necessary products for a hospital to function. The team at the University of Bristol has called their new cell line the Bristol erythroid line adult. This is also known as the BEL-A cells. The important difference between these cell lines and those that have been achieved in the past is that they have been able to effectively stop the cell in an early stage of development. This means that the early stages can then proliferate and divide into the later more mature cells. This is the important red blood cell, and they can do so on a large scale without dying. Unlike the earlier attempts, this can produce significantly more red blood cells and has the ability to be controlled and directed through use of signals. This is the combination of the molecular cell biology, understanding of the molecular pathways involved, and the ability to take a cancer cell line and then direct its development. The discovery was also made by another independent lab in Japan, and this was at the Nara Institute of Science and Technology. Both the Bristol team and the team in Nara are publishing their papers side by side. Although both developments are a long way away from producing clinically useful products, they are a significant step. The Bristol team claims that they intend to start development of clinically relevant products that can be put into trials in the near future. This whole process and creation has implications for patent and other legal ramifications that may limit the availability and the use of this particular piece of research, but that is a different matter. Thank you for watching. Please post any comments, questions, or suggestions you may have below.